Okay, Amy, so I think we're finally getting to the action here.
With the good bits.
Oh, the good bits. So how do we collect these samples?
Okay, so we already know what our question is, and we've already scribbled our quick map, and we've already randomized the map, and now we're actually going to toddle off, like we talked about, to those randomized grid squares, and we find the plant in the middle, and we've got our stuff with us that we're going to be collecting the sample with, and we've got our bag and our Sharpie for labeling.
Sharpie, yeah.
And so we find the plant, and then we're going to go halfway between the stem of the plant and the drip line. Now the drip line is not the drip line for the irrigation.
No.
Although I can understand why people would think that, right?
Totally.
Because we talk about drip lines all the time,
yeah.
and we don't normally talk about drip line in the sense that I'm meaning it now, which is like you've got your plant and it's got its canopy, and if it was raining, you know, the edge of that plant's canopy, there would be drips coming down. So that's the drip, hello darling, that's the drip line that I'm talking about.
Right.
So you're going halfway consistently between the stem and the drip line. Kind of important to go to that spot. And the reason why that's important is because the biology changes across the width of the root zone. So if we're always sampling between the stem and the drip line, we're always getting kind of an average reading. And so we're very consistent with that spot.
Okay, so that's the reason why with the drip line.
Yeah,
Okay.
that's why we would do that. And so what we're looking at doing is we don't really want the debris or the undecomposed organic matter on the surface of the soil. We don't really want that in our soil sample bag, because that's got other microbes doing their thing in there. So we're going to very gently scrape that off the surface. And if you're dealing with a lawn, there might be some thatch, some dead lawn that hasn't decomposed yet, which is a really good sign that you don't necessarily have the microbes you need if that thatch is still there. But you dig through it until you expose the surface of the soil. And that's where we start to take our soil core.
Now, can the soil be touched with our hands?
No.
Okay.
No touchy-touchy
No touchy-touchy.
with your hands. Yeah, and even when you scrape that organic matter off the surface, I would use your tool that you're collecting with or something, or even just put some gloves on, but just try not to be getting all your good microbes onto that soil surface when you're about to take that soil core sample. So don't touch it with your hands.
And then how about once you've got those cores and you put them in the bag, should you be kind of scrunching them up, let them be?
Oh, so tempting. You put it in the bag and you just want to
massage it,
scrunch it, and that's my job. I get to do the scrunching. But if you leave those soil cores in the bag just like that, the biology is a little bit more intact. Once you start the squeezy business, you're starting to spread things around a little bit. And then I would like to be the one that's doing that right before I check it under the microscope. So try and keep those cores. But sometimes you're working with a sandy soil or a dry soil that just kind of falls apart. Just let it fall apart, but don't assist the falling apart in the bag.
All right, no assisting. Okay. And so do we put all the soil cores in one bag?
We'll have talked about that usually beforehand. It really depends on your budget and what your research questions are that you're trying to answer. From most people, we're just trying to get the most representative answer we can in the smallest number of assessments possible. That's helpful for them and helpful for me too, right? So we just check it out, find our baseline, and then run with some management strategies. So usually we would put those 3 plants, the 3 cores that we took from those 3 plants, which would be 9, usually those cores would all go into the same Ziploc bag. Unless we were doing a formal research project or you had unlimited funding, we might check all of those 3 plants and say, oh, that's the one where the raccoon did whatever. That one we won't look at. We're going to take just these 2. But really, we're usually just putting them all in the same bag. And we'll have talked about that beforehand.
Can you tell me a bit more about the bagging? And should you, how much soil should you put in that bag? How much do you need for a sample?
Yeah, so we're talking about a clean Ziploc bag, and we want to make sure that we have at least 1/4 cup. If you've got 3 plants and you're doing 3 3-inch cores, you're going to have at least 1/4 cup of soil in that bag. But that's just enough that we can kind of maintain the conditions of the soil in a bag to get to me. The maximum, we don't really want more than 1/3 of the volume of the bag to be with soil, because we're actually going to leave that bag open, and we'll talk about that in depth, but we need the microbes to be able to breathe. We talked about how plants are obligate aerobes, and the microbes at their root zone also need air, and so if we were to fill a bag and zip it shut, we've just taken away the air, we've just changed the conditions for the microbes. So generally we're trying to get them to me in the exact same condition as they are in the soil.
Okay, got it. So now how do we go about in labeling the bag? I have heard that if you put a label in a bag, something might happen.
The microbes will eat it, and it will change the community dynamics. So unless that's part of your question that we're trying to answer here with the microscope, I would label the outside of the bag.
Right.
So you've got your Sharpie with you, and you obviously will put your name on the bag and the date it was collected. The time is important too, because generally I'm trying to get to these within 24 hours. With the microscope, and so if I know what time you took it, hopefully it happened on the same day that it came to me, then I can calculate that time frame, and we can be thinking about how representative things are. So your name, the name of the location, the date, the time you collected it. I think putting the outside temperature on the bag is helpful. If anything, if it just makes you think, gosh, it's 15 degrees today, I'm not going to put this in front of my heater in the car when I take it to Amy, you know, that kind of thing.
It actually can help kind of for next year's sampling as well, that if you wanted to stick with the same season and just to get a kind of idea of what the temperature was last time versus this time.
Fantastic point, Andrea. Yeah, that's great. And then maybe you've got more than one sample. So, you know, your name and the location might be enough if you've just got one sample, but if there's like healthy and unhealthy, or roses and geraniums, or carrots and squash, you know, if there are different ones, then make sure you clearly label which one's which on the bag. I do usually want to know what plant it is that you're trying to grow. Right. So if you've got soil that came from a raised bed that was bare soil, or a tilled field, something like that. I also want you to put your intended crop or the intended plant that you want to grow on the name of that bag there, just so that I can make sure that I'm reporting appropriately on those numbers that we see.
Cool. Anything else we should remember?
Just want to reiterate, don't squish the sample. Don't. That's my job. I'll do that when I receive it. Just try and refrain from that. And the other thing is, you know, I think next we'll talk about how to actually transport it.